Hello guys, I welcome you all to Biology Practicals. With me I have Pappu, and today you guys are going to have fun because we are going to do lots of experiment, starting with the first experiment that we have, and that is pollen germination. So let's get into it. So children, today we are going to see the pollen germination, and in this experiment we are going to do only few basic steps that you need to know. It's a very easy experiment. So it goes like this. The first thing that you would require is Vinca rosea. The flower looks like this. It's easy to observe, which is why we have taken that. And now we are going to use a needle, or you can also use a brush to shed the pollen grains onto the next thing, which is a glass slide. But it is preferred that you use a needle because since the pollen have the adhesive properties, it can go stick on the brush and it might be difficult for you. Okay, so now once we put the pollen grains onto the slide, we also need to provide the nutrient medium. Only then it is going to germinate. And what is that nutrient medium? It is going to be the sucrose solution. Post that, we would put a cover slip and we would observe it under the microscope. Also children, please make sure you would obviously be using your microscopes, right? And it is recommended to use the microscope under the power of 10x. So you do not need to look for 45x, just 10x would be more than enough. And I hope you have seen the microscope. It looks like this. So there is 10x power and there would be 45x. Go for the 10x one. And now we are going to see the experiment in real. Let's get into it. So first you would need the Vinca rosea flower. You can dissect it like this or using a needle. Then with the same needle we can shed off the pollen grains onto a very clean glass slide. And then with the help of a dropper, put 1 drop of sucrose solution. Post that, put the cover slip, and while putting the cover slip, make sure there are no air bubbles. And please wait for 10 to 15 minutes so that the pollen germination process can take place with the help of this nutrient medium. After that, you can view it under the microscope in 10x power and it will look something like this. Okay chums, so I hope you guys had fun doing that experiment. Well, now let's check out what could you actually see under the microscope. So yes, definitely so many pollen grains would be there, and some of them might have started the germination process, as in the process where they will be forming the pollen tube. So over here you can see tube-like structures being formed, right? So we say that these pollen grains which are there, they have started off the germination process. So this usually happens on the surface of the stigma. Once the pollen grain lands on the stigma, basically the stigma would be providing some nutrient medium for it such that it could grow the pollen tube. But as of now, we are not providing any stigma out there. We are providing a nutrient medium directly, and that is sucrose. So with the help of this nutrient medium, in the laboratory condition, it is going to start off the germination process. That is all about this particular experiment. Now, obviously, in your school, there might be different steps that they might have shown you. Please talk to your teachers who have done the experiment and then only go ahead. Okay, so the general steps which are mentioned in the lab manual, I'll be talking about them also. So according to the lab manual, it says that take a drop of 10% sucrose nutrient solution on a clean cover slip. So they are taking on a cover slip and they are asking you to dust the mature pollen grains of the Vinca rosea on this particular cover slip, and then to place the cover slip inverted onto what? A clean slide. Now that can be a little troublesome for all of us, which is why there was an easier way and we showed you that. Okay, and please make sure that you keep the pollen grains undisturbed for 10 minutes because obviously they have to take in these nutrients and go ahead with the process of pollen germination. I hope that is clear. Now, what will give you marks in this particular practical? You need to write some things up, some very important things, and I'm going to talk about that. But before that, I want to ask, haven't you subscribed to the channel yet? If at all not, then please subscribe to it right away for getting all the important videos. So the preparation of the slide will give you 3 marks. Okay, the experimental things that we showed you right now, if at all you do all of them correctly, you will get 3 marks. Now, you need to have 2 more marks because this whole experiment is totally out of 5 marks. So where will you get your 2 marks from? Listen to me. If at all you make this table for calculation properly, you will get 1 mark. What is this table about? So this table is about the total number of pollen as well as total number of pollen germinated. So suppose your slide looks like this. Okay, suppose
your slide looks like this, and this is your area where you are checking the pollen grains, and you have multiple pollen grains out there here and there. What do you have to do? Divide them into 3 columns, 1, 2 and 3. Okay, now check for the total number of pollen grains in this column number 1, column number 2, column number 3, because over here you have to write 1, 2, 3 observation. So in the first one I see the total number of pollen grains which are there, they are 3 in number. In the second one I see 1, 2, 3, 4 pollen grains are there, so I write it down. Similarly in the third one I see 2 of them are there. Now that is capital N. That is what? Capital N. That they are also asking you total number of pollen grains germinated. So you have to check now in each column how many of them are germinating. Suppose over here 1 is germinating, over here 2 of them are germinating, over here again nothing is germinating. So what do you have to do? Check for each of the column and write accordingly. So in column number 1 only 1 is germinating, so I write 1. In column number 2 I see 2 of them are germinating, so I write 2. In column number 3 none of them are germinating, so I write 0. Now what do you have to do? You have to do a total of this. So 3 plus 4 plus 2 is equals to what? 9. And over here 1 plus 2 is equals to what? 3. It would be equal to 3. You got the totality of it. If you make this table for calculation, you get 1 marks along with the content within it. Okay. Now, your next 1 marks depends on 2 things. One is the percentage of pollen germination. You have to calculate the percentage of pollen germination. Now, if at all you write this formula properly and you replace the terms over here within this particular formula, you are going to get your half marks. So, let me replace. So, small n is basically, so I have to write n is equals to 3, capital N is equals to what? 9. Now, you replace it. So, small n is equals to 3, 3 into 100 divided by capital N. What is capital N? 9. And you do the calculation. So replacement of all these terms is important, gives you half marks. Along with the formula also you have to write this down. Next is calculation of the pollen germination. So you do the calculation and that will give you what? Another half marks. Got it? So this is how it usually goes. So half plus half, 1. You already got 1 from here. So 2 marks done, and 2 plus 3 is equals to 5. This is how you are going to get your 5 marks in this particular experiment. I hope it's clear. Okay, do not worry if at all you do not see pollen germination. You can just write 0, 0, 0 out there. And also if you have any doubts, talk to your teacher also about it once before you enter. Okay, but do not panic. Works? So this is how it goes, my dear children. Also, one very important thing, I have already shown you the name of the flower in the video. It is Vinca rosae, and this is what you will get to see, the pollen germination happening. Also, one very important thing, we have as of now done the first experiment over here. So out of 4 you would be asked to do any one experiment, and you cannot choose that. It is totally upon lottery system. So we have done the first experiment on pollen germination, and now if at all you want to see the next experiment, do not forget to check our channel and do not forget to hit the subscribe button. Also, if at all you are a KCET aspirant, my dear children, I request you to go through ABHYAS KCET test series. It is definitely going to help you out. There are 35 KCET mock tests and also chapter-wise KCET PYQs, and all of it under proper exam-like condition. And all of it is for rupees 1499. The link will be there in the description section. Do check it out as well. Okay, so that's all for today, and wishing you all the best. And if you have any doubts, put it down in the comment section right below. Papu is super ready for the experiment number 2. I hope you guys are also ready. Now today we are going to do the experiment on pollen tube growth. So let's see what all are the required things. So first of all, my dear children, we all know we always require Vinca rosae flower. This is super important because we are going to take the stigma and style of this particular flower for the experiment. The pollen grains lands on the stigma and then forms the pollen tube, right? So which is why we need it. Now the second thing that we would definitely need is some hot water. So either in your institution they might provide you the hot water. I am not sure if at all you can see the fumes coming up, but it's there. Or there will be a burner and all the setups up there. You might have to heat the water and keep it ready. That's all. Now, going ahead, you will definitely need a glass slide and also a cover slip. And cotton blue is the stain that we are going to use for viewing it under the microscope. And we would need a watch glass for putting in the stain, and we would need a brush to transfer the stigma from the watch glass to our glass slide as well. So, and last but not least, you would also require the microscope. As I always say, please view it under 10x power. So let's go get ahead with this experiment.
Okay, so the first thing that you need is vinca rosea. You can dissect it using a needle, and this is what we will get. This is the stigma as well as the style. Now, we would be putting it inside some hot water so that the tissues become softened to take up the stain that we are going to use. So over here, with the help of a brush, transfer this onto a clean glass slide after some time. And at this point, if at all you want to cut off the extra portion, you can. And then we can add the cotton blue stain which is there using the brush or even a dropper. Further, it will look something like this. You need to put the cover slip on the top of it and then dab off the extra stain, and also make sure to press it gently and then view it under the microscope under 10x power.
Ta-da! I hope you enjoyed the experiment. Well, my dear children, this is how it is going to look under the microscope. So these are your pollen grains and they have the pollen tube as well. And the color is because you used cotton blue stain. Now, as I always say, wherever you are studying, they might be following some other procedure to do this particular experiment. So please coordinate with the institutions wherever you study. The common things remain the same only, okay? And according to the lab manual which is given to you, the steps goes like this. So I thought of explaining this as well. If at all someone has any confusion that, ma'am, why did you do it like this in our lab manual? It's something else. Don't worry, everything is the same only. The end result is going to be the same. So listen to me carefully how it goes according to your lab manual. So here they have taken a beaker with water and cotton blue stain and they had boiled it. So water along with cotton blue stain has been put and they boiled it. Okay, we did it separately, they did it together. Now, place the cut styles with stigma in boiling water for 5 to 10 minutes to soften the tissue. So why do you need to put the stigma as well as the style portion into the water? Because you want to soften the tissue so that this particular cell which is there or these cells which are there, they can take up the stain that you have mixed, which is what? Cotton blue. So in our video, we boiled it we put it inside the boiling water and then we took it and then we added cotton blue. But anyway, we did put in the hot water. Why? Because we wanted to make sure that the cells are now softened. They can take up the stain which we are going to put. Now, after that, what we did in the video is that we put a bit more stain and basically we put the cover slip properly and we viewed it. And before viewing it, basically we gave a press also, right? A gentle press was needed over there and then basically we viewed it under the microscope. Now over here, some people, okay, they also put a drop of glycerin. So after taking this particular thing out from the mixture of water as well as cotton blue, so it has got the stain, okay. Now you take it out and you can put a drop of glycerin. Why is it being done? So glycerin prevents drying up, okay, it prevents drying up. So that is the only reason why we use glycerin over here. Some people do, some people do not. And then after putting the glycerin, you can put a cover slip and then gently, okay, gently press on the material. Don't do like this, otherwise the cover slip is going to break up. You would not be able to see anything, okay, you would have to repeat it again. Otherwise, you can also use the back side of the pencil and gently press it up with the help of that. Okay, and if there is any stain towards the side, please remove it with the help of blotting paper as well. Now after that, you can directly view it under the microscope, and as I always say, 10x. Okay, so one more line is written: if you fail to observe pollen tubes, mount another stigma, observe the slides under a microscope. So either way you have to observe it. Now one very important thing, the extra portion of the style you can cut it off as well. Okay, after putting it in the hot water and when you take it on the slide, you can cut it off and then add glycerin and whatever thing has been done, you can do that. Okay, similarly in the video that we did also, you could have cut the style portion, the extra portion, and then you can add the stain. That would be better. After putting the stain, if you go and cut, it might be a bit troublesome for you. Okay, so yes. Now one very important thing, you do not get marks just by doing this. By doing this, by preparation of the slide, you get 3 marks. But now still you need to have 2 more marks. Where do you get that from? Let's go ahead and check that. But before that, have you subscribed to the channel yet? If not, subscribe to it right away, my dear children, because we are doing so many things for you, right? So we deserve some form of love in the form of subscriptions as well. Okay, now coming over here, you need to also make diagram. When you make the diagram, you get those 2 marks. And this diagram needs certain labelings. Okay, certain labelings such as stigma, style and the pollen grain and also the pollen tube. So first of all, make the pollen grain and label it. Make the pollen tube and label it. Label the stigma, label the style. Each of them carries half marks. Half marks. Okay, so half into 4 is basically is equals to what? 2 marks. So you will get your 2 marks out there. So this is the experiment for pollen tube growth, and this is how you are going to get your 5 marks out of this. And my dear children, so with this we are done with the second question also. So as I always say, out of these 4 questions you might be given 1 to do. Okay, so we have already done it in the previous video. If you have not watched it, go watch it. And now in this particular video, we did the pollen tube growth experiment as well. Now the next video, we will be seeing the number of locules and the type of placentation in hibiscus or China rose. Also, if at all you are someone who is preparing for KCET, do not forget to enroll into ABHYAS KCET test series. It's going to be a really helpful test series for you because we have got 35 mock tests. There is chapter-wise KCET PYQs and all of it in exam-like condition. In 60 minutes, you have to solve it. And this is the most trusted test series as well. We had so many, rankers out there from this test series. So we recommend it to you. If you need it, definitely check it out in the link given in the description section below. Wishing you all the best, and that's all for today. See you soon with the next experiment. So by seeing Papu Vera hibiscus, I'm pretty sure you know which experiment we are going to do. We are going to see the T.S of Ovary. In the case of hibiscus, we have to make sure to understand how many locules are going to be there in the ovary. So let's get ahead with this experiment. And for this, obviously, the first thing that you would need is a hibiscus, or you also call this as China rose. And you would require a watch glass with water. If it does not have water, please fill it up, that's all. And you will require a brush for transferring purpose. You would require a blade to cut off very thin slices of the ovary, that's all. Now I'll tell you how you have to do it, and we'll definitely require a glass slide, and this time you do not require a complex microscope over there. There's a simple microscope that would be needed, and it would be enough. And there is no need of cover slip as well. So let's get ahead with this experiment. So the first thing, we have to take out the ovary, and how do you do that? You just pull it off like this, and if you notice inside there is a small ovary. I'm not sure if at all you are able to see this, but it will be there. So the thing is, you just need to pull it off like this. Fine. Now, just a second, we need to. So now we have it. The ovary is right over here, it's small, and you can just discard the tip right there. We do not need that. So let's go ahead and discard it. We need to make sure we are just taking thin slices. Let's go ahead and get it up. So here we have got, I am not sure if at all able to see this, inside the water, right? Then you have to select the thinnest of all of them, because you would be able to view it even better with that. Take a glass slide. We need to transfer this up onto the glass slide. The purpose we put it inside the water is that so that it does not dry out, and while transferring it, make sure you drop in some water over here. Otherwise, again, there are chances that it might dry out. So now it looks like this. You do not need to put any cover slip for viewing it. And also clip it so that there is no disturbance, and view it in this. Yippee! We did that experiment as well. And by the way, if at all you were not able to see the ovary, for which there are high chances, let me tell you where is it located. So if at all you go deep over here, you would be able to see or locate the ovary right over here. So the flower, the hibiscus that we used, it was not mature one. So the ovary was literally very small. But if it was a mature ovary, you could have seen basically a little more larger one. Yes. So this is how it goes, and I hope you got it. So rather than pulling it off like this, there is also a way where you can use a blade or a needle to cleave this open and then take this particular portion out. So that can also be done. So accordingly, as per your institution, please go ahead with this particular process. Okay. Now, how it is going to look? It is going to look something like this under the microscope. And my dear children, over here, how will you get your marks? So 3 marks are for the preparation of this particular slide. Okay, so you got your 3 marks. After that, you also need your 2 marks. 3 plus 2 is equals to 5. This is totally out of 5, right? So how do you get that? 1 mark is for reporting the number of locules. And it's pretty simple. Locule is basically the ovarian cavity that you have, okay, the parts of the ovarian cavity that you have. And over here you can see there are certain ridges in between. This is a ridge, this is a ridge, this is a ridge, this is a ridge, making the ovarian cavity convert into 5 parts. I'll say that it was a normal locule out there, but now it is converted into 5 locules, 5 different parts. So this is known as pentalocular condition or 5 locular condition. So this is a locule, this is a locule, this is a locule, this is a locule, and this is also a locule. You have 5 of them, and my dear children, you would also get your marks for the next thing, that is by reporting the type of placentation. So placenta is something that is going to provide nourishment to the ovule which is there. Now depending on the location of placenta, there is different type of placentation. So over here, if at all you see, the placenta is in the middle, and you also have certain ridges being made. This type of placentation is known as axial placentation. What do you call it? Axial placentation. So this particular placenta is supporting the ovules. These things that you see is nothing but ovules. And per locule, you would be having almost 2 ovules up there. Okay, generally. So that is something that you need to remember. Remember, over here you have, and also it formed the ridges. This is known as axial placentation. So 2 things, if at all you write, you get your marks. I'll write it down over here. The number of locules that you have to write is pentalocular condition. Pentalocular condition. The second thing is basically reporting the type of placentation. As I told you, it is axial placentation. So this carries 1 mark, this carries 1 mark, you got your 2 marks, and that's how you will get your 5 marks as well. And my dear children, if at all there is someone who is watching the video and feels like, oh, it's a good video, then the first thing that you have to do is click on the subscribe button, because my dear children, we will be bringing up a lot of things for your PU2 board examination as well, for your KCET examination as well, for your COMEDK examination as well. So do not miss out on this. And by the way, on which experiment are we in? So I told you, out of the 4 experiments, you would have at least 1 experiment to do. Okay, according to lottery system, you would have to pick 1 experiment, and it's not of your choice, obviously. So you would have to do it. So you need to know all these experiments. So we have done a video on pollen germination. If at all you have not seen it, go watch it. We have done another video on pollen tube growth. Go watch it. And now we are on this particular thing where we have to make a temporary slide of the transverse section of ovary and report this particular thing. So we have done that, and now we will be going ahead with the next experiment on nucleic acid staining. So stay tuned for that. It is going to come up really soon. Okay, now if at all you are a KCET aspirant and you are wondering where to study from, we have brought the ABHYAS KCET test series. Make sure you have a look at it. Check out if at all you think it is useful for you or not. The previous years, so many of them got the best use of it. We got rank 171 out of 3 lakh children. Okay, rank 443, 693, and the list goes endless, my dear children. So please check it out. Over here you will get 35 mock tests. There will be chapter-wise KCET PYQs as well, and all of it in real exam-like condition. So 60 question under 60 minutes, you have to do it. So for further details, the link will be there in the description section. Go check it out. Wishing you all the best and lots of love to all of you. I hope you are enjoying it. If at all you are enjoying, do let me know in the comment section. And if you have any doubts, also do let me know in the comment section below. See you soon with the next experiment. So today we are going to play with fire, is it? No, you're not going to play with any fire. This experiment is also a simple experiment where you have to stain the nucleic acid, and it goes like this. So let's go ahead, let's see how many things do you require for this. So over here you would be requiring the onion peel. You might get it exactly like this, or probably you might have to take it up. And my dear children, you would require a watch glass containing water, because after taking the peel we are going to put it inside the watch glass so that it does not get dried up. Post that we will also require the glass slide as usual. Onto this glass slide you are going to transfer the peel and you are going to put acetocarmine stain onto it. Okay. And my dear children, after that you can put the cover slip, dab off any extra stain which is there with the help of a blotting paper, and then you would have to use the spirit lamp for heating it up for only 10 to 15 seconds. Now for this you will require a holder so that you can hold the slide as well. Okay, and then we have to view it under the compound microscope, and make sure you view it only under 10x. Let's go ahead. So my dear children, today we'll need onion peel. Slowly try to peel off the innermost skin and then put it in a watch glass containing water. This is to make sure that it does not dry up. Further, with the help of a brush, transfer it onto a clean glass slide and make sure that there is no folding. And then you can use a dropper to put a drop of acetocarmine and further warm it up over the spirit lamp for just 10 to 15 seconds. You can put a drop of acetocarmine again and then later put the cover slip and further view it under the microscope and make sure it's under 10x power. And ta-da! We did this experiment as well. And my dear children, you would be observing something like this under the microscope. So as you can see, there are stacks of cells which are arranged very close to each other. And this red or pink color thing that you're able to observe is nothing but your nucleus. And what did you do to make your nucleus glow like this? You used a stain which is known as acetocarmine. This whole particular experiment is on nucleic acid staining, right? And nucleic acid such as DNA, RNA, everything is found inside what? Your nucleus. And you wanted to stain that. Okay, your DNA is negatively charged and I'll say it is acidic in nature. So for staining this up, you need a basic dye, and that basic dye is what? Acetocarmine. You know, positive, negative, they attract, which is why acetocarmine is able to stain this nucleic acid up, and we were able to see this under the microscope. So let's see what are the general steps that have to be followed according to the lab manual which is given to you, which is uniform to all of you. Now obviously there are certain steps which you do differently in your institution. Please do it in the way your teacher have told you itself. Okay, now coming over here, let's see the general steps. First, peel off the epidermis from the scaly leaf of an onion bulb. So first thing, we have to peel it off the epidermis from the scaly leaf, and then what we have to do, we have to place it on the slide and we have to add few drops of water. Why? Because water prevents drying up. Okay, water prevents drying up. Now take out a small piece of the epidermal peel. Now suppose over here in this particular video it was already small, so you do not need to cut again. But suppose you are getting a bigger peel, so you have to make sure that you cut out only a small peel out of it. Wipe out the water carefully using the blotting paper out there, and then you have to add 2 drops of acetocarmine. Again, you can use a dropper. In this particular video we already had a dropper like thing at the top, which is why I used that. Now over the epidermal peel and heat gently on a spirit lamp. So you have to heat it gently on a spirit lamp for almost 10 to 15 seconds. That's all. Why are you heating it up? Because you want to soften the tissue. You want to soften the cells so that it takes up this particular stain, that is acetocarmine. Okay, it's a plant cell, and plant cell has cell wall, a lot of things. So you want to make it soft so that it can take things up. Now, apply a cover slip over the epidermal peel, avoiding air bubbles, and wipe out the excess stain with the help of a blotting paper. Yeah, and then observe the prepared slide under the microscope. Now, this will give you 3 marks for sure. Again, you will need 2 marks more for getting 5 marks. And how will you get that? You have to do 2 things. First is reporting the shape of the cell. So we say the shape of the cell is what? Rectangular. Even though it's not exactly rectangular, but we say it is almost rectangular in shape. So please write this. Now, reporting the number of nuclei per cell. So as you can see, in this particular cell you just have 1. Similarly, in another cell you just have 1. What do you call this condition as? You call this condition as uninucleate condition. Uninucleate condition. If you write this, you get 1 marks. If you write this, you get 1 marks. 1 plus 1, 2, and 2 plus 3, 5. You have got your 5 marks in this experiment as well. Got it? It's that simple. Now, one very important thing. Have you subscribed to the channel yet? If not, Baba, subscribe to it right now. Because ultimately, we will definitely help you in 110 ways possible by us. So please trust us and keep on trusting us if you already do, and lots of love from all of us. Okay, now one very important thing, my dear children. So in the first question, as I told you, you had 4 experiments. You need not do all the 4 experiments. According to the lottery system, you would pick a chit, and whatever experiment you have got, you would have to do it. So I have already done the video on pollen germination, pollen tube growth, as well as the T.S of Ovary. If at all you have not watched these videos, go watch this, because by luck you might get anything out there. So do not take any risk. And also over here we have done the nucleic acid staining experiment. With that we end up all the first question, all the questions which were there under category 1, and all of them will give you what? 5 marks. Okay, now we will be going ahead with the next question, which is common to all of you. Everyone has to perform this experiment, and that is the experiment on mitosis. Now this experiment, people think it's very, very difficult. Trust me, it's not too difficult. Okay, and PU2 board has made it a bit more easier for you, and I'll be talking about that in the next video, so stay tuned for that. And also, if at all you are someone who is preparing for KCET examination, go check out our ABHYAS KCET test series. It has 35 mock tests, and there will be chapter wise KCET PYQs. This is something that we have added up newly, and there will be questions along with some proper timing. So it's like exam like conditions would be maintained so that you have an understanding of your speed and accuracy. If at all you want to know more about this, definitely check it out in the link given in the description section. And my dear children, it's one of the most trusted test series out there, and since you guys asked it, we launched it a bit more earlier this time. Okay, so check it out if at all you think you need it. And that's all for today. Wishing you all the best. Lots of love from me. Bye-bye. Take care. Today we are going to play with some hydrochloric acid. But don't worry, it's dilute hydrochloric acid. So everything is pretty much safe. My dear children, we are going to do the mitosis experiment where we have to spot the stage of mitosis and we have to report that. Now, this experiment is something which is compulsory for all of you to do. There is no lottery system up over here, which means that we have to be pro at it. So let's see what is this experiment all about and what are the requirements for this particular experiment. So the first thing, you would need onion root tip, because we are going to observe the stages of mitosis in the onion root tip. This is the stored one. In your school, things might be different. Please check with your teacher. Now going ahead, you will require dilute hydrochloric acid. This is for softening up of your tissues out there, and yes, and it makes sure that the cells which are there, it takes up the stain. What is the stain we are going to use over here? We will be taking up acetocarmine stain over here. Now you would also require watch glass. Okay, and onto this watch glass, what we are going to do is, we are going to take the onion root tips, we are going to put it over here, and then we will add dilute hydrochloric acid along with what? Acetocarmine. When we put dilute hydrochloric acid, it makes sure that the stain has been taken up. After that, we are going to heat it. How? With the use of spirit lamp. Now again, for holding it, we have a holder as well. Okay, we'll just heat it for 10 to 15 seconds, that's all, not more than that, and we will keep it aside and we will cover it as well so that it does not dry out. Post that, we are going to take that particular specimen, and then we are going to put it on the actual glass slide where we will further do the experiment. Okay, I hope it makes sense, and you would also require the brush for the transferring purposes, and you will also require the blade as well. You will see why is it required in this process when you see this experiment. So let's go ahead.
So firstly, you need to take a few root tips onto a watch glass. I have taken only one, but you can take more, and then add few drops of acetocarmine stain. This is very important. And the same place itself, you need to add few drops of HCl and heat it up with the help of a spirit lamp for 10 to 15 seconds. Then keep it aside for 30 minutes and make sure you cover it up with the help of a Petri dish. Further, after 30 minutes, take this up onto a clean glass slide. This is the root tip that we need, so cut off the extra portion. Further, add few more drops of acetocarmine and then put the cover slip and dab it up with the help of a blotting paper so that it removes the extra stain, and also press it gently and then view it under the microscope at 10x power.
And finally, we did this experiment as well, and my dear children, this is what you will get if at all you do it properly. Okay, so you would be able to observe multiple stages of mitosis, starting with the first stage, which is prophase. So prophase, this is a cell, and over here you can see that everything is clumped up in the center like this. So this means that the chromatin basically which is there, the chromatin fiber which is there, it has condensed and now it has formed the chromosomes. Apart from that, the nucleus membrane which is there, nuclear membrane which is there, it also starts breaking down at this particular point. So prophase, it looks something like this. Now metaphase is the next thing, where in the equatorial plane you would be able to see all the chromosomes are lying up in the middle. This is metaphase, M for middle, you can remember like that. Now in anaphase, they will start moving away from each other. Okay, in anaphase you can clearly see there is a movement on both the directions out there. Now telophase, it is kind of not very easy to spot, but just understand the chromosomes which are there, actually very easy if at all you get an understanding of it. So the chromosomes basically move completely towards two different poles. So they will be there towards two different poles. So anaphase looks like this, where they are still separating and they are moving away. You can clearly see they are moving away. Over here they have completed the movement, they are towards both of these ends. So this is what you can observe. Now as I always say, in your institution you might have had different steps, but there are certain steps which are already given in your lab manual, about which also you should be having an understanding, and please go according to the teacher who have taught you. Okay, general thing that I want to tell you. Also, 3 marks for preparation of this particular slide. Rest 2 marks is still remaining, so listen to me very carefully. First of all, let's see the general things which are there. Take onion bulb and place it on the test tube or glass bottle filled with water so that the base of the onion bulb touches the water. So basically over here they are telling you how to form the onion roots. So they are telling you either you can take the bulb and put it in a test tube containing water, or put in a glass bottle containing water. The tip should be in touch with the water so that it will be growing up. Now after 5 days the roots start growing. Cut few root tips and transfer to a vial containing freshly prepared 1 part of glacial acetic acid and 3 parts of ethyl alcohol for 24 hours. Why are you treating it with glacial acetic acid and also ethyl alcohol, my dear children? Do you know that? Do you know? So glacial acetic acid makes sure that basically any sort of cell division would be stopped. Okay, all the cell division process which is there, it should be stopped, and that is why we use glacial acetic acid. Obviously there is more to it, but as of now this is a general level of understanding that at least you should be having. Now why do you use ethyl alcohol? So that the extra water content which is there in the cell, that can be removed. That is why you use ethyl alcohol. So both of them are mixed together, and basically you put this root tips into that. Okay, this process is known as fixation, and then preserve the root tips in 70% alcohol so that no microbial growth and anything can take place. Now this thing you do not have to do. Okay, the people who are there in the laboratory, they might have already done, they might have already prepared the root tips as I have shown you in this particular video. So it was already prepared. What you have to do starts from this point. So while preparing a squash, take 2 to 3 onion root tips into a watch glass. Take it into a watch glass. Okay, take 2 drops of 1 normal HCl and 3-4 drops of acetocarmine stain. So basically you are adding acetocarmine as well as HCl. What does the HCl do over here? HCl is also used for softening up the tissue so that it can take up the cells can take up the stain, which is your acetocarmine. Now, you are going to gently warm the watch glass over the spirit lamp. Again, this is for softening. Okay, so that it can take up all the stain out there, and once the fumes start coming, close the watch glass with a petri dish. So make sure you do not have to wait for that generally. So basically after doing it, after heating it for like almost warming it for like 10 to 15 seconds, you can take the watch glass and basically cover it up with the petri plate. Okay, so that there is no drying up happening up there. Now after some time, the root tips, take the root tips onto a glass slide and then again add 1 to 2 drops of acetocarmine stain. Then you can place a cover slip and gently press the cover slip as well. Okay, with the back of the pencil, or you can also press with the help of your thumb finger. But you have to do it gently. Don't do it like this and break off the cover slip. It might be too much trouble for you. Okay, so the basic thing, as shown in the video also, acetocarmine plus HCl, you have to put it in the watch glass containing the root tip and then warm it up and then bring it up, cover it up with a petri dish, let it be there for some time. Then after some time, okay, after some time, take some root tip out there. If there is any extra thing out there, cut it off and then just put 1 to 2 drops of acetocarmine and then cover it with cover slip and then view it under the microscope. Okay, so this gives you 3 marks. Now what about those 2 marks out there? Those 2 marks you will get by identifying any one of the stage. So as of now, PU2 board has said that you do not need to identify any stages. But if at all in your school it's been like that, so please identify. But generally please ask, okay. Generally it is said that you do not have to identify, but at least basically writing about a stage would be really, really good. And also you have to comment on the stages observed. So basically any 2 points you have to explain over here. It will be really good if at all basically you explain, suppose you're taking anaphase, explain 2 points about anaphase, or metaphase and explain 2 points about metaphase, but please do that. You will get your 1 marks with that. Okay, so this is general idea. If at all your teacher have told you something else to do and that's how you will get your marks, please do that. Okay, so according to institution it might vary, but this was the general thing which was there. So previously you had to identify one stage and allegedly you had to comment on the stages observed and you have to write 2 points about it. But this time some things have changed up. Talk to your teacher also. Okay, so only the bottom line is that you have to be prepared with 2 points of each phases, know about it, okay, and please go and write it there. You will get your marks. Okay, and if you have any doubts regarding the 2 marks given over here, ask to your teacher at the earliest. Cool, so that works, I hope so. And also, if at all, okay, one more very important thing. The prophase, as I told you, it looks like this. Metaphase in the equatorial plane, anaphase it is started separating, and telophase, my dear children, they are moved apart and they are at both the terminals out there. Cool. By the way, if you have not subscribed to the channel yet, subscribe to it. What are you waiting for? Subscribe to it so that you can get all the updates. And my dear children, did you like the video? Are you liking all the experiments which are being done? If yes, let us know. Okay, let us know through your comments, through your likes, through your shares, and also if there is any doubt that you need to ask us, please ask us. So in the previous videos that I did, I had completed these experiments. So there was question number 1 where you had to pick one experiment and do it. It's not like you get to pick it. It's lottery system. Basically, with whatever chit you are picking up and whatever experiment you have got, you would have to do the experiment. So there was one experiment on pollen germination. I did that. There is pollen tube growth experiment. I did that. I also did the transverse section of ovary in hibiscus. I also did the nucleic acid staining. That was also done. So if at all you have not watched those videos, please watch because you will get 5 marks out of it. Now the next question which is there is this. Make the temporary slide of the given material, that is onion root tip, and over here you have to talk about the stages of mitosis. So this is the thing that I did right now. There are few more things and that comes under spotting. It comes under spotting. So please stay tuned for it. The other videos will also be coming up. If at all you want anything specifically, you can let us know in the comment section below. Okay, so that's all for now. And also, if at all you're someone who is preparing for KCET, definitely check out the ABHYAS KCET test series, one of the most trusted test series. The previous year, we got so many children who got great ranks. Obviously, their hard work plus little bit of our hard work also. We had Prajwal with rank 171, Kiran with rank 443, and the list goes endless out there. Okay, so there are 35 mock tests over here, and there will be chapter-wise PYQs as well. And all of it in real exam-like conditions. So 60 minutes, 60 questions, that's all. So test your speed and accuracy, and you also get to understanding of how well are you performing in KCET. So please check it out. The link will be there in the description section below. Wishing you all the best and lots of love from Rasul ma'am. Please take care of yourselves and ask your doubts in the comment section. Bye-bye.